This is Module A4, uh, Objective 4. We're going to look at the characteristics of the muscle fiber and identify all the components of the sarcomere. When we look at the muscle fiber, and again, let me remind you that the muscle fiber kind of translates into cell, and they always put that word in quotation because it's very different from what we would consider a cell. A muscle fiber is a very long, multi-nucleated cell. So even though we use the word muscle fiber, do realize that this is the individual muscle cell, no matter how long it may be, and again, it can be up to 12 inches or so. The muscle fiber is covered on the outside with a structure known as the sarcolemma. And this is nothing more than the cell membrane of the muscle fiber itself. And here we can identify the sarcolemma. And the sarcolemma surrounds the sarcoplasm. And again, this is nothing more than the cytoplasm that we've seen before. Put this down as cell membrane. So this is the overall outward, at least, and inner appearance of the muscle fiber. As we look at the sarcolemma, we notice these small channel openings along the surface of the sarcolemma. And these are the openings of the transverse tubules, or T-tubules. And the function of the transverse tubule is to carry the action potential. And this is a term that you will come to know a great deal about. This is nothing more right now than an electrical impulse. And the T-tubules will transmit the electrical impulse or action potential through the cell. And we note that the little openings of the transverse tubules are located all up and down the muscle fiber. And the reason that this is so is when the muscle fiber contracts, we need to have the entire unit contracting at the same time. We find that the T-tubules have the same properties as the sarcolemma, so the same, they're made up of very similar structures, and we find that the action potential, again, or electrical impulse, or nerve impulse, reacts the same as it runs down the sarcolemma or as it runs down the T-tubules. And just again to point this out, we can see here that this canal is going to continue as it runs through the muscle fiber. The myofibrils, these are subdivisions of the muscle fiber. This is an entire muscle fiber here. Again, we see the sarcolemma. I don't see any T-tubules right now, but they're in there as well. And these lengthwise subdivisions are the myofibrils. And the myofibrils are composed of mainly two protein types. We have thin filaments, and thin filaments are made of the protein actin, and we have thick filaments made of the protein myosin. And we'll talk more about actin and myosin and their interactions shortly, but for now, just associate the thin filaments with actin, the thick filaments with myosin, and that these proteins are found within the myofilaments. The sarcoplasmic reticulum is a membrane-bound organelle that is very closely related to the endoplasmic reticulum. And the sarcoplasmic reticulum helps to transmit that nerve impulse. And what we find is that these sac-like pouches hold a structure, or basically hold calcium ions. As we look at the overall structure of the sarcoplasmic reticulum, and again, it's all of the blue in this figure out of your text, we have these sac-like pouches, and as we notice, the sarcoplasmic reticulum kind of expands into very small pouch-like structures closely associated with the T-tubules. And we'll talk about the importance of that a little bit later, but these chambers are known as terminal cisterna. Terminal cisterna. When we have one T-tubule and two terminal cisterna, we have effectively a triad. Here's the term and the figure, and this includes, again, one T-tubule and two terminal cisterna. And the function of the triad, and namely the terminal cisterna, is to hold and concentrate calcium ions. They actually have little pumps. If this was the membrane-bound sac, we have small ion pumps that, through the action of ATP, we actually get calcium ions pumped into the sarcoplasmic reticulum. And the calcium is just going to be stored. We mentioned when we were looking at bone that calcium is the most abundant ion in the body, or mineral in the body. And I also mentioned that calcium plays a very pivotal role in muscle contraction. In fact, it's the calcium that actually initiates muscular contraction. So the role of the sarcoplasmic reticulum is to hoard and store that calcium so that whenever muscle contraction is required, whenever a signal is sent from the brain to contract the muscle, that calcium can then be released, and again, that's going to initiate muscular contraction. It will initiate muscular contraction. This is just an overview. We see here the muscle fiber, and we know it's a muscle fiber because we see the nuclei, the multinucleated appearance. And basically, we're blowing up this region here where the myofibril is being pulled out. And we see all of our structures. Here's the sarcoplasmic reticulum. We have the triad, which again is the T-tubule and two terminal cisterna. We see the sarcolemma. We see the entrance channels to the T-tubules. We also see the sarcoplasm. So as we pull out the myofibril, again note we see the presence of thin and thick filaments. And again, these filaments are different protein types. Thin filaments are actin, and the thick filaments are myosin. I'm going to stop midway through this objective to give everybody a chance to absorb and try to memorize some of the material that we just covered. We're going to be moving into some very detailed specifics of the sarcomere, and you really need to understand the anatomy that was just introduced before we move on. So the last slide that I have here is the same slide we just looked at, the same figure we just looked at, minus all of the labels. It would probably serve you well to print this out and to go through and label all the parts of the muscle fiber discussed thus far. So we'll return and review this material before moving on to the more detailed specifics of the sarcomere in the next section. So there'll be a part 2 to this module objective.